definitely go through the exam, and there should be comments on your individual questions about reasons why you didn't get full credit, if you didn't for a particular response. I also posted a key, so you can look at that as well to compare your response to the response that was anticipated. Otherwise, I think many, I don't know how you, but the grades were very good. I don't think we had a single perfect score on the first exam, but there were a number of them on the second. Yes, question? Go. The other announcement that came out is with regards to the lightning talk slides. I've had a few students asking questions about what the expectations are for those. So I did something I should have done before, and I added details to the actual instructions of that assignment. These lightning talks are supposed to last about 2 or 3 minutes. So the general rule of thumb in giving a presentation is to have 1 slide for every minute you'll present on. And so I've stated in the instructions you're allowed a maximum of 4 slides. You don't have to include all 4, but one of them needs to be a title slide. So just with your name and title on it. And then 2 or 3 more slides that feature some image from the paper you're summarizing that will allow you to explain aspects of the paper well. One rule of thumb, whenever you present a figure, make sure you can explain it all the way through and illustrate the axes, if those are indicated and so forth, if it's a graph, but just explain them well. You can have text on slides. Of course, it's okay to have text. Question?
Yeah, are we assuming that we all know the knowledge that was presented in this class? For example, like if you're presenting on squids, you need to specify the anatomy of like mollusks, you need to talk about the difference between arms and tentacles. Like, or are we going into it saying, oh, we've covered this, now I just have to discuss the different, like the things that the paper talks about that we haven't.
Yeah, that's a good question. So you know, who is your audience? I think your audience is for the fellow students in the class who have engaged in the material during the course of the semester, so they should be familiar with some of the themes that you're presenting on. So good point. So the audience is us. And remember, the whole point of these is for us to share, for you all, to share your summaries. It gives the rest of the students, as well as me and the GSIs, an opportunity to learn too from what you uncovered in your research summary article. We've got close to 70 students in the class. We'll hear close to 70 different summaries of research articles. So it's going to be it's going to be exciting. With regards to text, one recommendation I have is it can be tempting to write out whole lines of text on your slides. But then you end up saying either the same or something equivalent. So an audience member has the option of either listening to you or reading your slides. And what I usually do is read, because oftentimes I can read in my own head and it's easier to read than to listen. So using little bullet points is a good strategy in talk. Why bullet points? So there's enough there for you to remember what you're talking about, then you can elaborate on that point. This exercise is what, 5% of the total? It's worth 5% of the total grade for the entire assignment. So I'm not going to be evaluating you on your public presentation skills or the excellence of the slides or anything like that. Most of you will get 5 points out of 5 awarded for completing the assignment on time and everything like that. But that's the expectation. I think there was another point I was going to make, but I don't remember that now. Any questions about lightning talks or the slides? Feel free to come to office hours or to ask me questions via email if you have a particular question about the material. Yeah, 4 slides, maximum 4. One has to be a title slide, your name and either the citation for the article you chose or the title of that article, and then grade your choice. Do you have one with a picture of the study organism? And so forth. Any questions related to that? Okay, so last week we almost made it through the lectures on mollusks. We left off with bivalves. You all had the opportunity to see various bivalves in lab late last week. Before I complete lecture on mollusks and discuss bivalves, are there anything interesting that you learned about bivalves or experienced with bivalves that you'd like to share with class? Because I have one. Yeah.
I've been cut by the shells a lot of times whenever they're cracked and broken in lakes. So
Been wandering around the lake, and there is, we're going to talk, so it's freshwater lakes around here?
yeah.
Yeah, so there are a variety of unionid mussels that can be common in lakes and rivers around Michigan. I'll talk about them at the end of the lecture, end of the lecture on bivalves. One of the species is actually, they have all kind of weird names. Warty back and so forth. But one of them is actually named the heel splitter. And precisely for that reason, it has an edge that juts out from it, and it can split many a heel. Okay. And we often see many shells of freshwater mussels around. Freshwater mussels, though, are protected. And so to do any kind of work or research on them, you need a special collecting permit. So actually handling unionid mussels is a no-no. Collecting them, I'm sure mistakes happen all the time, especially with young children picking up some unionids and bringing them back and putting them in an aquarium and so forth, but they are protected species because they're threatened for a number of different reasons. Any other cool observations, experiences, or something interesting you learned from the course reading about bivalves? Well, the one thing I wanted to share, because going through the lecture today, it dawned on me that I'm not talking about one of the most interesting features about the bivalves, and it pertains to a condition that is known as DUI. Everyone know what DUI is? Not driving under the influence, but it is doubly uniparental inheritance. Doubly uniparental inheritance, DUI. What do we know about mitochondria and mitochondrial genomes? They're inherited from the mother. So you have your mother's mitochondrial genome. I have my mother's mitochondrial genome. We all have our mother's mitochondrial genome. Within many groups of bivalves, and based on the distribution of this trait within bivalves, it may be an ancestral feature of bivalves. Many freshwater bivalves, including unionids, a variety of different species of unionids, so not all have this condition, where it's doubly uniparental inheritance. Females pass their mitochondrial genome to all of their offspring. So males and females inherit the mitochondrial genome of the mother. But males pass their mitochondrial genome and their mitochondria to their male offspring. And so the mitochondrial genome of the male can be detected in gonad tissue, where gametes are produced, the sperm are produced, but no female mitochondrial genome in that tissue. Whereas in the rest of the tissues, the somatic tissues, somatic cells of the males, those mitochondrial genomes are maternal, from the mother. So a male is heterogametic. It possesses mitochondrial genome of both its father and its mother, but in different tissues, whereas females possess mitochondrial genomes of only the mother in somatic and gonad tissues. It's an odd situation that is unique to bivalves. So in thinking about today's lecture, I recall that this is one of the most interesting, not one of the most interesting, but another interesting feature about bivalve biology that I neglected to include in the lecture. So I wanted to comment on that before the lecture. So it's a good thing that bivalve lecture got delayed. So bivalves are things like scallops that we saw in the class, with little white spots surrounding them, variety of different freshwater mussels. Those are unionids, oysters, clams, and so forth. They go by a variety of different names. Cockles are a name given to certain types of bivalves. So many different types of bivalves. It's the second largest group. There are around 15,000 described species. There are a variety of different marine and freshwater forms. In comparing them to other mollusks, they are interesting or distinct in terms of the fact that they do not have a distinct head region. They have a region where the mouth is, but nothing else predominant about that region. There are some exceptions, of course. There are some snails that don't have a radula. There are some apocophorans that have a radula, but no bivalve has a radula. And then their mantle cavity, in comparison to other groups, is greatly extended and encompasses almost all of the living animals. They're also distinct in that they have this two-valve condition. Now, thinking about the anatomy of a bivalve, it has these two valves. The foot extends anteriorly towards the front of the animal. So if we were to open up the valve, the mouth would be in this region. The anus is in the posterior region. This is the dorsal surface. Growth continues. You can see these growth lines anteriorly, posteriorly, and ventrally. The earliest shell is recognized in adult individuals. That's this umbo region. And so then the shells open ventrally with the hinge dorsally, and they have right and left valves. So looking at this from cross-section, there's a right valve and a left valve, right valve and a left valve. Vast majority of bivalves, not true for all, there's some oysters in which the valve that is used for cementing to a hard substrate is a different size than the other. But most bivalves tend to have symmetrical shells. The right is a mirrored image of the left. Interior of the shell often can tell us aspects about the biology of these animals, where they live. There are various muscle scars and attachment scars. These are two areas where adductor muscles attach from one shell to the other shell, and they leave a little scar. Based on the size of the scar, you could evaluate how much force is needed to close those shells. There's also an attachment scar for where the siphons, incurrent and excurrent siphons occur. The depth of this scar can usually indicate how large those siphons are. With a mollusk or a bivalve shell, these two shells, there's a small proteinaceous material that's attached to the hinge region, the place where the two shells are connected. They're connected via this ligament. And the ligament, if you were to relax a bivalve to where its muscles are not contracting, the valves naturally gape and open. So here the adductor muscle is relaxed, the valves open. A recently dead bivalve, I don't think we had any of these in class, but they will tend to gape because they're no longer in control of their muscles, they're dead, and the hinge causes the two valves to open. So, as a bivalve, how do you close your valves? By contracting those two muscles, the adductor muscles, anterior and posterior adductor muscles, to close the valve. If you want to open it, just relax those muscles and the valves open. This is different. We're going to talk about brachiopods today. Brachiopods are another group of animals that have two valves in their shells. They could be similarly sized to bivalves. These, though, do not have left and right valves. They have a top and a bottom valve. So their orientation is distinct from those of bivalves. Moreover, where the valves attach at the hinge, for these brachiopods, the ligament does not force the two valves open. Instead, there are two sets of muscles. One set that closes the valve, just like bivalves have. The other set works to open the valves. The other difference, and you can see this here, is that the valves, dorsal and ventral valves, in the case of the brachiopods, are different sized. So they are not symmetrical. They are not mirrored versions of each other in most cases. Most bivalves bring in water into this large mantle cavity, and there are cilia associated with the gill filaments that are responsible for moving that water, and they're also taking food particles that are present within the water and then moving those food particles to the mouth. Internally, again, they have a digestive tract that flows through the body, terminating in the posterior region by the excurrent siphon. See the cross-section of the adductor muscles. They have a three-chambered heart. The gills are, in most taxa, used for dual functions. For filter feeding, which most bivalves do, there are a few bivalves that use the siphons instead to deposit feed, but many are filter feeding, vast majority are filter feeding. They also use it for gas exchange. The gills are vascularized and gases are removed as well as brought into the body through the use of the gills. lots of bivalves have symbioses with other animals. You look, I think we've all seen images of giant clams. How is this, the orientation of this giant clam different than what we were just talking about? It's upside down. Not only is it upside down, it's gaping. You are in, these typically occur in coral reef environments. You're snorkeling in one of these areas. They're typically gaped, wide open. They're not closed and protecting the valves. You bother a mussel, and it's gaped, it's going to close up. If it recognizes you're around, they're going to be closed. These are open. Why? Because they house zooxanthellae, and not just giant clams, but other clams. Anyone remember who zooxanthellae are? Yeah.
They form a symbiosis with corals, right?
Exactly. So the zooxanthellae corals, the reef-building corals, have zooxanthellae. When they get bleached, it's because they're losing their zooxanthellae. The zooxanthellae are unicellular eukaryotes. They are dinoflagellates, and these giant clams house zooxanthellae in their tissues. That's what's giving particular colored tissues that we can see within this gaping clam. They are, I've never seen one this large in the wild. They tend to be fished for their meat. It's supposed to be very tasty. And they're also used in the aquarium trade, so forth. So there are various attempts throughout the world, tropical parts of the world, to grow these and replenish reefs with these. The tissues of these giant clams are interesting in that where you see the algae housed in the periphery of these tissues, they are surrounded by special cells, iridocytes, that are iridescent cells that are associated with kind of the focusing of light energy into these regions, presumably, that's what this paper suggests, while also affecting the transmission of it so that they aren't so damaging to the housed algae. So they're supposed to be making use of the energy while reducing photo damage that would occur within the algae themselves. We talked about chemoautotrophic bacterial symbioses with annelids. Here are some of those cyphaloblinids, organopherans. Here, there are also some mussels present, some clams, some other mussels that have similar associations with chemoautotrophic bacteria. Instead of the trophosomes, where the bacteria occur, these clams and mussels house the bacteria within their gill tissues. There could be a number of different types of symbionts. This particular figure has a cross-section of a gill, and they're using this approach called FISH, fluorescent in situ hybridization, essentially taking a tissue, taking probes that are specific to either the host or particular symbionts, and seeing where they occur. And you can see that the different symbionts are intermixed within parts of those gill tissues. We didn't see any of those in class, but when we did see in class, we had shipworms. These consume wood. I don't know if we'll talk about termites later, but termites are another organism that consumes wood. They don't do it all on their own. They, like shipworms, have symbiotic bacteria within their guts that help to break down the cellulose of the wood. So lots of different symbioses within bivalves. Finally, unionids and freshwater mussels that are common around us here in Michigan. Unionids, there are over 1,000 species worldwide of unionids. It's a rather species-rich group. There are about 300 species in North America. Used to be much more abundant than they are today due to impacts on the environment, damming of rivers, and even utilizing shells for production of buttons in the 20s and 30s. There's been decline in population sizes of many of these unionids. There are species that have gone extinct in North America. And they're an interesting bivalve that occurs in freshwater systems in North America, but they also have very interesting life cycles. Most bivalves tend to be dioecious and spawn gametes. Fertilization takes place in the water column, and then the larvae ultimately settle down. Trophophore, velager stage larvae. Within unionids, in this case, the females don't release the eggs. They take up sperm from a male, or from multiple males in some cases. The eggs get fertilized, and then larvae, the embryos, are brooded within, and they may be, like in this example here, released from the female. And then the glochidia, these are little tiny glochidia, on gill filaments of a fish host. So these are somewhat parasitic larvae in terms of they latch onto gill filaments and then ultimately release and settle down and form an adult. So these take advantage of fishes for some nutrition aspects as well as for potential movement. Some images of glochidia attached to gill filaments. They can be detrimental to the fishes in terms of their blocking the filaments, and so they are not allowing for efficient respiration, gas exchange. So not all unionids, though, just simply release their larvae into the water column. Some package larvae into these particular structures, ovisacs, sacs of eggs or sacs of embryos, that take a variety of different forms. Some of these little red spots into eye spots that look like maybe little larval fishes. Others that look like larval stages of various arthropods, crustacean insects. And these then are released, and they have little sticky parts they may attach to substrate or a rock. And then if there's fish comes by to investigate, these break open and release the various glochidia. One example. Another example is with other unionids that have extensions of the mantle that may resemble potential prey of fish. This may look like a little fish. With the fishes, these some are fish-like, some with there's worm-like projections that are designed as lures for attracting prey, hosts of their larvae. Another interesting one is this particular species here that has these strong hook-like regions on the posterior margins of the shell, and they will be gaped, and a fish may, I should say gaped this way, a fish may come to investigate, and then it closes the valves and actually latches onto the fish, and then larvae can be pumped into the fish for continuation of the life cycle. So a variety of different means, both to attract prey using lures and structures that, these ovisacs, that look like potential prey items of the host fishes. Some species we know there's a specific host fish for the particular species. In other cases, they may rely on a variety of different hosts, so it's not host specific. Any questions about bivalves or mollusks in general? We talked about all 8 classes. There's at least one extinct class of mollusks that I know of, but we won't go into details here. So what I originally planned for today was talking about the last 6 Lophotropozoan phyla to address. There are 6 more to address. They're all included in a single chapter, although the relationships of these are not necessarily so clear, or putting them in one chapter is not necessarily the best thing to do, but that's how our book does it. So what I first want to do is just a final lecture on Lophotropozoans, talk about relationships of these to other Lophotropozoans, and see the different interpretations about how these are related to each other. Then we'll talk about the biologies of these different groups. Before we do that, I have one interesting feature thing that I read in the book, see if anyone else uncovered that. But something interesting you learn about today's phyla from the chapter reading, I forget which, chapter 8, was it chapter 8? Something interesting you read about them in the chapter? Or some, if you've had any unique experiences with a sickly offering, that would be cool if somebody had a unique experience with a sickly offering. But if you had a unique experience with any of these groups or something else you learned about them in another class or so forth. Something. Nobody has anything interesting? What is the longest, what is the longest animal?
Longinus with 60 meters in length, although it's only like 5 to 10 millimeters wide.
Yeah. So, cool, you got that too. Ribbon worms are one of the groups we're going to be talking about today. And most ribbon worms are, they can get fairly long. I think most of the largest ribbon worms I've seen, maybe about 10 inches, something like that. But our book discusses one that is 60 meters long. What's the longest other animal that we know of that's living today? Which group of animals would we think would be the biggest living today? Which phylum? What? Mammals? Yeah. Mammals. And which mammals are so big? Elephants get pretty big, but some of the biggest are whales. I think the blue whale. And I looked it up because I saw it 60 meters, and off the top of my head, I have no idea how large the largest whale is. But blue whales tend to be the largest. But blue whales, at least what I read, they tend to get a maximum length of 30 meters. 60 meters. That's about, almost 2/3 of a football field. That is huge. And twice as large as the largest other mammal. So that was my largest animal. So that was something too. That was something I wrote down because it's like, wow. And I read the text every time I teach the class, and you know, I think I forget about that right here, that these ribbon worms can get so huge. Anything else from the readings that you did? Yeah.
It's more like an observation. I think I was a little surprised that like sometimes when I was looking at like some of the animals kind of remind me of like Bacterial, and I was like, these are so far apart, but they look like similar. Like they have very similar structures in the olive phase, right? Like I don't know how that like I don't know what the reasoning is for that, but I just found that very surprising.
Yeah, so, and those are interesting phenomena when we think about the evolution of different organisms. The fact that we have similar body types, I mean, the fact that entoprocs and ectoprocs, which have these individual zooids that have these little feeding tentacles, very reminiscent of hydrozoans. And it suggests that particular feeding mode, if we think of all possible different types of life forms there possibly could be, that's something that's evolved multiple times. I doubt they derived from a common ancestor that had that. So it tends to suggest that particular mode is advantageous. And so I think, I know there have been people who have tried to speculate on what life would be like elsewhere. That's probably a good prediction. Somewhat filter feeding like colonial organisms that we see in cnidarians and some of these Lophophorata taxa. Anything else? Okay. So the 6 groups that we'll discuss are the phoranids. This is a relatively small group in terms of number of species present within it. There's only about a dozen or so species that have been described within it. They are worm-like organisms with a crown of tentacles, what's called a lophophore. These are hollow tentacles, extensions of the coelom into them, reminiscent of cnidarians. They occur in little chitinous tubes. They very much resemble polychaetes, but have distinct feeding structures, are not segmented, and so forth. Ectoprocs, this is kind of a bad image because it doesn't show the feeding structures, but this is a colony made up of individual zooids, each with a lophophore, one of these feeding organs, crown of tentacles in each of the individuals. So these are individual specimens. This is a colony of individuals. Ectoprocs are another group that have these feeding tentacles surrounding the mouth, feeding tentacles surrounding the mouth, feeding tentacles surrounding the mouth. The Brachiopods, who we introduced just a few moments ago, that have these 2 valves, they also have a feeding organ that is made up of tentacles that are extensions of coelom, contain extensions of the coelom into them, so they're hollow tentacles. I should say, we're talking about the lophophore. Extensions of the coelom within, and they are also ciliated. The cells of cilia on the lophophores. So if we're thinking about cnidarians, and sometimes if you were off and you're sampling something that you're not sure, is it a cnidarian or is it a ectoproc or an entoproc? Looking at the tentacles and seeing whether or not the tentacles are ciliated will at least give you some information as to whether or not they're a cnidarian or another phylum. Nigeria. Tentacles are not ciliated. We talked about all of these groups, and then another are the Cycliophorates. So are the rotifers, circle-bearing, wheel-bearing. These have a circle of not tentacles surrounding the mouth, but cilia that surround the mouth. These are very minute animals. These we can actually see with the naked eye. These much less so. So there's a variety of different groups. And then the group that I haven't discussed, Technomeridians. They do not have any tentacles, regular tentacles. This chapter is somewhat of a grab bag for ribbon worms being placed with these other groups that have a ring of cilia or a ring of tentacles surrounding them. But part of the rationale for the authors of our text putting them here is some molecular phylogenetic results supported the close affinity of these to some of the other phyla represented here. And that is shown here in the tree from our text that I've adapted from what our text shows with Cycliophorates, these small little beasts with cilia, ring of cilia surrounding the mouth, are apparently closely related to entoprocs based on molecular phylogenetic investigations. And the ectoprocs, which are these that have a lophophore, which these also have, and these also have a lophophore, right? Feeding apparatus. In terms of its ring of tentacles, extension of this coelom, ciliated. All of these have a lophophore-like structure. If you get into the nitty-gritty detail of it all, though, lophophores of ectoprocs and brachiopods are similar in that the cilia, the acilium, emerges from a single cell in the epidermis of the tentacle. So they tend to be uniciliated cells. Whereas, I just messed that up. I should have put these together. Phoronids and brachiopods have uniciliated cells on the lophophore. Well, ectoprocs, they're multiciliated. So the lophophore is exactly the same in these as it is in the others. So ectoprocs have a unique lophophore compared to brachiopods and phoronids. And then this feeding structure of the entoprocs, if you get into a strict definition of it, the strictest definition of a lophophore is a feeding structure, as I described, the mouth in the circle of tentacles and the anus outside. That is met by the phoronids, the ectoprocs, and the brachiopods. Literally, ectoproct. Proct. We used this somewhere before. Proct is another term for anus. Ectoproct, outside anus. These two literally are ectoproctic in terms of the anus is outside of the ring of tentacles. Entoproct, you can guess from the name, inside anus. The anus is in the interior of that ring of tentacles. So literally, if we accept this as a literal definition of a lophophore, then entoprocs do not possess a true lophophore. It's a similar structure, but it's presumed to be uniquely derived. And those who examined the development of lophophores of ectoprocs compared to brachiopods and phoronids have said the same about these because they have the multiciliated cells in the lophophore. So before molecular phylogenetics came out, there were some ideas about how these things were related based on the structure of Lophophore and where the anus occurred. This is how our book does it, which does follow some molecular phylogenetic results. Ectoprocs are not closely related to the others with the true Lophophore. Entoprocs that have a Lophophore-like structure, but it's not true because the anus is within it, are separate from those as well. Close relationship between Cycleophrans, which have this ring of cilia, and Entoprocs, which have a Lophophore-like structure. This is supported, and this continues to be supported by molecular phylogenetic results. And in some results, it suggests that Nemerdians are close relatives of these. I don't expect anyone to remember the tree I showed you last week. Oh, before I can go there, let me summarize. So we have Polyzoa, that includes these three clades, Trochozoa, which includes annelids, mollusks, and these other three phyla, which are grouped together. Brachiopods and phoronids as supposed sister species. This is maintained. Most molecular phylogeny support that relationship. What I learned about, and I was sitting in New York City many years ago, was that we have Lophophorate taxa, Brachiopods, phoronids, and ectoprocs, and actually entoprocs. These are all assumed to be members of the same clade. There weren't distinctions made about position of the anus or about multiciliated versus uniciliated cells in the Lophophore. Most connotations, though, of today don't recognize the entoprocs as a member of a clade known as Lophophora, true Lophophore taxa. This, again, is what our tree from the chapter represents. Monociliated cells of the Lophophore are unique to here, but we find that Lophophore evolved in ectoprocs independently, and entoprocs have a unique structure that isn't turned to Lophophore, but that resembles the Lophophore of other taxa. It's a ring of tentacles, ciliated tentacles, that surrounds the mouth. But you can see what supports these groupings that our text follows, and our text even, I think the authors even say in the chapter, we're not sure, but this is what we're currently going with, based on results from some molecular phylogenetic investigations. I think it was specifically a paper from 2013. Okay, this is where I was leading towards. Last week on Wednesday, we were talking about, or even Monday, we were talking about the position of where mollusks fell out, and this is the most recent molecular phylogeny I gave, showing where it potentially falls out. This also supports the true Lophophorate phyla as sharing a common ancestor. So in this superphylum referred to as Lophophorata, containing bryozoans, who are the ectoprocs. I didn't say this, but bryozoa is another term. Anyone know the root of that? Bryo? What's a bryophyte? Somebody knows plants very well. The bryophyte is a moss. So a bryozoan, so a bryophyte is literally a mouse or a moss plant. A bryozoan is literally a moss animal because many of these are erect forms. They look like moss. So bryozoa is another term for the phylum Ectoprocta. Nemerdians in this particular reconstruction are close relatives of annelids. They're not segmented, but they presumably possess the common ancestor sometime in the past, or at least that's what this particular tree supports. And then entoprocs and cycleophrans, again, we have a ring of tentacles on a true Lophophore in the entoprocs, and the ring of cilia in the cycleophrans that are closely related to each other. So if we go back to what our book presents, slightly different. Instead, these three species share a common ancestor, and annelids tend to be close relatives of Nemerdians, the ribbon worms. This is a unique feature within ectoprocs, but otherwise a lophophore presumably evolved from the common ancestor of all three groups. So, contrary to how our book presents it, I'm going to be discussing first the three Lophophoric taxa. I don't have any reason, the most recent phylogenetic investigation supports this, but I don't have any reason to suspect that this is definitely true. Additional phylogenetic investigations may overturn the close relationships inferred for these three taxa. But they all do possess the same type of feeding structure. Entoprocs, we're assuming that the Lophophore-like feeding structure was independently derived in them, though it is similar in structure to the Lophophore of these other taxa. Ciliated extension of the coelom, but apparently uniquely evolved within the entoprocs. So the Brachiopods kind of introduced them already. Brachiopod recognize pod from foot, and brach literally refers to an arm. So this term tended to come about from the fact that the Lophophore, based on early interpretations, was a modification of a foot-like structure. It's an armed foot. It's these arms that gives rise to tentacles, but it's derived from a foot. And this is because originally they were thought to be mollusks. Hell, I think if we were an early naturalist exploring different organisms, bivalves, true bivalves are plentiful, we come across a brachiopod that has two valves, typically formed from calcium carbonate, reminiscent of the bivalve shape. We would assume that these are relatives as well. They're also commonly referred to as lamp shells because some give the appearance of being a lamp. They are marine, relatively shallow water. There are around 300 species known today. So, not a very diverse group today, but if we go back to the fossil records, there are around 30,000 species. These are all extinct, and most existed during the Paleozoic. They went extinct. A number of these lineages went extinct at the end of the Paleozoic. They have, like with bivalves, they have two valves. So they are a bivalve group. Mostly, and the two shells are top and bottom, dorsal and ventral. So that's the orientation of the body. There are some where they are more symmetrical than others. There are two main groups of brachiopods, inarticulate and articulate. Articulation refers to whether or not there are teeth present, how the two valves articulate with each other. Articulate brachiopods have little teeth, like many bivalves do, associated with that hinge area. And we can guess that inarticulate have no teeth associated with that hinge area. Which major difference between these and bivalves, aside from their planes of symmetry and their asymmetry of the shells themselves have to do with they either attach, they all either attach through cementing, they attach to a substrate through cementing of one valve, like what oysters do, or the vast majority have this structure that emerges from one of the valves, referred to as a pedestal, that will attach to a substrate. So these are attached bivalves. In modern species are rather nondescript, but some of the fossil taxa were greatly ornamented and very distinct shapes to them. Here's a picture of one that washed up on the shore and others that could be found underneath a rock and in the tidal area. So they can be relatively common. On initial inspection, I think most people, even many good zoologists, would think, oh, it's a clam, it's a mussel. Differences, again, would be, for instance, a pedestal for attachment, differences in the feeding structure, a Lophophorata instead of a gill. Oh, reminder. There's another scene from the Natural History Museum, at least the old one from one of the dioramas they had. There were little models that were contained within them. Brachiopods were very abundant and diverse during Paleozoic times. I think this is a scene from the seafloor. Here's a representation of what these brachiopods, and that's an actual specimen of what these models are supposed to represent. So very rich in brachiopods diversity. Anyone ever go to the state park in Petoskey looking for Petoskey stones? Petoskey stones are hard to find, but I'll give you, if next time you're there, you can find brachiopods in between. So they have, it's from the same Paleozoic era material. There are brachiopods that have broken free of the stone that they were embedded in, and so you can find the shells, plenty of brachiopod shells around there. They're much smaller. But if you want to find something, if you aren't having luck finding Petoskey stone, you can find some brachiopod fossils. So their history, great diversity during the Paleozoic. And the Permo-Triassic event was a major extinction event with lots of different marine animals going extinct or suffering to a great extent as brachiopods did during that event. Looking at brachiopod diversity through time, I think this looks at number of genera, this looks at number of families. During that Permo-Triassic major extinction event, brachiopods nearly were wiped off the earth, and they've only maintained much smaller diversity through the Cenozoic. Bivalves, on the other hand, weren't all that greatly affected by the Permo-Triassic extinction event and have continued to diversify through time. We can see that their greatest diversity, 40 families. At the Permo-Triassic event, these had nearly 70 families. Much greater diversity of brachiopods than bivalves in the Paleozoic. Any question?
Yeah, why? They're very similar body structures. You expect that the external environment would have similar effects on both groups. So what is it that was in bivalves that allowed them to have a great diversification while brachiopods were immediately or almost went extinct?
That's a good question. That leads up to a topic I want to discuss. Because many people in the past looked at these fossil records and said, look what happened. I mean, certainly if you look at bivalves, they've been diversifying since their origin. They weren't more diverse than brachiopods. But what happened after the Permo-Triassic event? In the Mesozoic, bivalves diversified tremendously. Some people have speculated that there may be similar trophic levels that these two different groups occurred in. And with For whatever reason, brachiopods went extinct. We don't necessarily, at least I don't necessarily know why they did. But with their extinction, this may have opened up niche space, available resources for bivalves for them to diversify. And that's certainly a hypothesis. But I think many people have pounded it into the dirt to say that this may not be related. And one fact is looking at this, they show a constant rate of increase. Rate of diversification does not tend to be related to how diverse the brachiopods were. They continued diversifying at their rate, even when brachiopods went extinct. So I know Stephen Jay Gould, who was a famous evolutionary biologist, he used to write a number of popular essays. And one of his essays he wrote probably back in the 80s, 1980s, was something like 2 ships that passed in the night, because he was arguing against the adaptive explanation that brachiopod extinctions triggered the diversification of bivalves. Instead, it's more than likely that this had no impact on that. And even though he raised this 40 years ago now, good Lord, I've seen recent publications by early career paleontologists who are revisiting this theme to try to evaluate whether or not there's any relationship between the decline of the brachiopods and diversification of the bivalves. A little bit about, so they have various internal organs associated with their viscera that's located in posterior part of the shell. The anterior, much of the anterior region is occupied by the Lophophora. And one of the other differences that I forgot to mention when we're comparing brachiopods and bivalves just now, that I discussed earlier when we were talking about bivalves, they have sets of muscles. You can see that if this muscle contracts, what's going to happen? It's going to cause it to open because it's located way at the base of that valve. So contraction of this muscle causes it to open, while contraction of this muscle will cause the valves to close. So it's a different system than we find in bivalves. Bivalves rely solely on the use of their ligament for opening. You relax, you open. These, they would have to relax one muscle and contract the other to open. They have horrible terms like there's still a mantle cavity where the lophophore is present in. It's a mantle that produces the shell tissue. Some of these are remnants from the past when brachiopods were considered to represent mollusks. Brachiopods, so the articulate and inarticulate groups have a lot of features that differ between them. One of those differences is with the morphology of the larvae. Within the articulate, I'll show this little movie here because you can see some swimming larvae. With regards to the articulate brachiopods, their larvae are reminiscent to the trochophore-like larvae that we saw in annelids and mollusks. And they metamorphose into a juvenile stage that has a lophophore, while the inarticulate ones have these little larval stages that already possess a lophophore. So there's very large differences between articulate and inarticulate brachiopods. Most brachiopods also are dioecious. They spawn their gametes. Fertilization takes place externally. And then larval stages that live or exist for a particular period of time before they settle down. For major differences between the types of larvae by inarticulate and articulate brachiopods. The ectoprox, literally outer anus, for the position of the anus relative to the lophophore, the anus emerges from outside the Lophophore. I will use the other term, moss animal, bryozoan, quite a bit. That's how I originally learned this file. And as you can see, others have too in that phylogenetic results paper. They refer to it as bryozoa. There are marine and freshwater forms. Freshwater forms tend to have unique aspects to their morphology. There are around 4,000 species. Most are marine. They can have a variety of body plans. They could be erect. They could be branching. They could be these huge, these are freshwater bryozoans, huge gelatinous mass. They could be encrusting, getting this membranipara with the calcareous test. There could be these types that are colonial and occur like little tiny caps. These are little tiny things about what's smaller than a dime, not half the width of a dime, that sit on muddy seafloor bottoms. So they have a variety of different types of morphologies. They're a fossil record because many of these had good skeletal features, that hard skeletal features that preserved. They have a very rich fossil record. Going back to the Paleozoic, there are a number of different groups that existed, and many of these went extinct during the Perma-Triassic event. Some of these extended into, diversified, and then have continued to today. Others originated more recently and are found in the Cenozoic. So there, like with many groups we've talked about, there are extinct groups that no longer are extant. That's the definition of extinct. And then the current diversity is based on only a few groups that originally diversified within this file. They are one of the classic examples of punctuated equilibrium. Some of you have evolution. You should remember what punctuated equilibrium refers to. This is a hypothesis put forth by Stephen Jay Gould, the other person, and Niles Eldridge, both paleontologists. Gould was the one who talked about two ships passing the nights, Brachiopods versus bivalves. The notion of punctuated equilibrium is that there are periods of morphological stasis within a lineage, and then periods of morphological change occur only during speciation events. Why? Well, you may, because of the incompleteness of the fossil record, if this is, say, time, and this is morphology, kind of shown in that diagram over here, we may see a situation where we go back and characterize morphology of a particular species. It falls into this general range, and it's from this particular time. We go to another time in history, we see what may be the descendant of that lineage over here, and then we see somewhere later in time, morphology here. And one of the interpretations, which were original interpretations, is that evolution is gradual. And it's incomplete, so we don't have the individuals here. But if we did, they would be somewhere between these different morphologies. That was the notion of gradual evolution that Darwin put forth, Ernst Mayr, and others have discussed that evolution tends to be gradual. Gould and Eldridge came up with an alternative hypothesis that instead of it being gradual, what we may have is a lineage existing in time. There's a speciation event from which another lineage, and so it may represent a situation where we get abrupt change associated with speciation events. And so it's another way of explaining a fossil record like I showed here, that's incomplete. It could be gradual, but it may not be. And this is actually from an evolution text that shows aspects of morphology on the x-axis and time on the y-axis, and different species shown in different colors here, and inferences about different species, no way of testing that, but very dramatic shifts, some species with different morphologies coexisting at particular times, so it's suggesting that this may be a situation where organisms are maintaining stasis, they're not changing aspects of, in this case, their morphology over long periods of time, and when they do, it's when speciation event occurs. So, bryozoans have an extensive fossil record. Patterns like this can be identified from it. In terms of the morphology of the bryozoans, it's somewhat reminiscent when we talked about Hydarians. They have these zooids that are the individual members of the colony. They occur in a skeleton that is secreted by them, the zoecium. The living part of the animal consists of the lophophore and the aspects of the internal, the gut, and so forth. Again, the anus exiting outside of the ring of tentacles, the mouth in the middle of that ring of tentacles, and then they're interconnected through a shared body wall and this skeletal matrix with other individuals that are developed, like with Cnidarians, through budding. So an individual buds off individually. If you haven't seen the video at the beginning of class, it showed an amazing video showing a larva develop into a single polypide, not polyp, but polypide, and then giving rise to additional individuals through budding. There are other groups. This is one of those calcareous ones that encrusts on giant cup and so forth, in which there may be a membrane that is covering them, membranipara. So this membrane covering the individual compartments where each polypide resides. They have little doors, just like the doors on the Gasteropod shell, Vibraculum, that can be closed so they can withdraw the lophophore and close the lid for protection. Most of the individual zooids are feeding type zooids. Again, like we talked about with hydrozoans way back in January. There could be individuals of the colony that are specialized for particular processes. There are these that are modifications of the zoecium that are used for keeping things from settling on the colonies. They're known as Obicularums, avi, because they're kind of bird-like. They look like a little bird beak that works to keep things from settling on the animal. There are others that are long, like materials, modified from the operculum of the original zoecium, that are known as Vibraculum, that again function in keeping things from settling on the organism, on the colony. You can see some of these little Obicularia on different parts of this particular colony. There are individual zooids where eggs are brooded. The beginning video that we saw showed that, the emergence of an embryo, a larval stage, from an individual oecium, where the eggs occur, are fertilized, and then they develop, and then are released the adult. Most ectoprocs tend to be dioecious. I'm saying one thing, thinking one thing, and saying another. Most ectoprocs tend to be monoecious, either simultaneous or going from one to the other. you could have so individual zooids, though, tend to either produce male or female gametes if the entire colony is monoecious. Different types produce different types of larvae. There's a lot of variation in larval types that come from ectoprocs. This is one that is considered to be trochophore-like of this particular bryozoan. These trochs that are on it siphon up larvae from membranifera. It's distinct, although it has these rings of cilia. And then one of the interesting features, and this we'll see how far back your memory goes, is that some freshwater bryozoans will asexually produce staboblasts, where there is an outer protective covering. There are typically two valves also that cover these. Some of the floating forms may have these hook-like features on them. And these are overwintering stages, so they're produced asexually. I was reading a report back in the 1930s, that somebody measured the production of statoblasts in Douglas Lake up at the biostation, and I think they had something like 100,000 per every square meter where these bryozoans were occurring. So these could be very numerically abundant, produced during the fall months. And conditions improve in spring, they then will develop into a single zooid and ultimately a colony of these. So where did we hear about something like this very similar before? Yeah.
Sponges?
Sponges and the gemmules, exactly. It's very, I don't think we get confused with the gemmule, but it's a very analogous feature that is observed within temperate bryozoans, freshwater bryozoans and freshwater sponges, because they can't survive during the winter. All right, well, bivalves got me behind, so we're not able to finish all of today's lecture. I don't want to start talking about phoronids. We have phoronids, singleopterans, entoprocs, and nemertians still to go. So we'll cover them on Wednesday. We'll also talk about ecdysozoans, nematodes, tardigrades, I think everything except for the arthropods of the ecdysozoans. So we cover a number of different phyla this week. The remaining weeks will be arthropods. Arthropods is the largest assemblage of animals, and so we're taking multiple lectures to go through them. We'll actually have 2 different labs on the arthropods. This week's lab will feature Lophophorate taxa, no ribbon worms, no Cicleopterans, unless somebody wants to bring in a lobster, and nematodes, and their kin. All right.